transmission and or conservation of genetic information. So they basically want you to know, how does the structure of DNA make it an information molecule? Do you guys remember? What is it about DNA and those nitrogen bases that makes it really good at storing information? Remember anything about that? Remember we talked about it's kind of like words, right? We use letters in a specific order to give us words, right? And we said that the order of the letters is the information. And so in DNA, it's the order of the nucleotides that codes for the proteins. So let's go ahead and mark this up a little bit. First of all, a little bit about the structure. Remember the term double helix. DNA is a double helix, if you could underline that. Okay, that says a lot. First of all, double means that it's made of two. So if you underline the word two, it's made of two polymers that go in opposite directions. And so if you want to, over here on this diagram, let's number them. There's one, there's two, right? So there's two polymers, and you guys know from geometry maybe, or you'll know from geometry eventually if you haven't taken it yet, that parallel means that they are side by side and never intersect. Okay? When we say helix, helix means that it has a spiral shape, right? Spiral shape, it's a twisted ladder. Think of it as a twisted ladder. If we untwist it, it looks like a ladder. If we twist it back up, it kind of looks like a spiral staircase, if you've ever seen one of those, okay? All right, the structure of DNA is kind of like a ladder, right? It looks a little bit like a ladder, where in the middle, right, we have nitrogen bases. You guys remember? Those are the nitrogen bases. And there are weak hydrogen bonds that hold them together in the middle. And so when we say nitrogen base, does anybody remember any of them? Adenine, thymine, guanine, and cytosine. Those were the four nitrogen bases. Remember, maybe we'll label that. And there, that could be G or C or A or T, right? One of the four, right? Does anybody remember what the P stands for? P for phosphate. Phosphate. And then what about the S? Does anybody remember what the S is? Close?
Sugar.
Sugar. Right, you got it. Oh, there's Kenny. Sugar. So let's label the S. Sugar. Okay. And then maybe one last important detail here is that the sides are where the covalent bonds are found. So let's label the thing holding the sugar to the phosphate is a covalent bond. Is that a strong bond or a weak bond? Strong, good. And that's important because we don't want these to get out of order. We want them to be in the right order. What do we call it if they get out of order? You guys remember? It's called a mutation, right? Mutation. That messes up the information. Remember, we also talked about how DNA is able to copy itself, right? And we said it happens in 3 steps. Let's go ahead and number them. 1, 2, 3. First of all, the DNA has to unwind. Or maybe we could even say, if we think of it as a zipper, whoa, it has to unzip, right? It breaks those hydrogen bonds down the middle, and we can see that happening over here, right? It's unzipping. Then free-floating nucleotide base pair. Remember, A with T, G with C. What is it? Apples in the tree and cars in the garage. And so let's maybe draw a little arrow. A goes with T, G goes with C. Apples in the tree, cars in the garage. And then lastly, DNA polymerase comes along and forms bonds between the nucleotides on the new strand. We think of DNA polymerase as kind of like a stapler. It holds these nucleotides together and makes the new DNA molecule. Go ahead, Jenna. What's up?
In other words, for G and C is carrots in the ground, and in other words, for A and T, avocados in the tree.
Avocados in the tree, right? Yeah.
Yeah, because
we don't think about avocados
grow in
enough,
trees, and carrots grow in the ground.
Okay. At the end. Oh, yeah, they're wonderful, and they're supposed to be pretty good fat for you. Pretty good source of fat. Okay, at the end, DNA replication is semi-conservative, right? Semi-conservative means that each new molecule of DNA is half original and half new. And remember, this makes it very accurate, very good, accurate process, because each half of the new DNA molecules was the original molecule, right? And so they have to contain the same order of nitrogen bases for that to work. Okay, any questions here? All right, let's just continue on. We'll go on to number 2, genetic engineering. We're just gonna knock this one out because it's kind of short. Remember, we said genetic engineering, the anchor for this says, explain how genetic engineering has impacted the fields of medicine, forensics, and agriculture. For example, selective breeding, gene splicing, cloning, genetically modified organisms, and gene therapies. And so let's just remind ourselves what some of these different techniques are. First of all, engineers are problem solvers, right? If you're an engineer, you solve problems. There's different kinds of engineers because there's lots of different kinds of problems in the world. And so a genetic engineer is somebody that solves problems using DNA as the solution. And so the first thing we'll talk about here is DNA fingerprinting. You guys remember we did that little activity with DNA fingerprinting? This allows us to compare the DNA found in evidence at a crime scene with suspects. And so if there's any biological evidence like blood or skin or hair or whatever, saliva, we can compare the DNA in that biological evidence to the DNA of a suspect. Do you guys remember this? These are called DNA fingerprints. They're kind of like barcodes for people. So DNA fingerprints, think of them as like barcodes. Everybody's DNA makes a different pattern of bars. And so if you look at this gel electrophoresis, whose blood matches the blood stain? Who is it? John, you're right. And so John and the blood stain, are a match. And so that means that that blood came from John, okay? Any questions? Okay, so that's something where we can use genetic engineering to help solve crimes. Let's go to the second page here. The second one is genetically modified organisms. These are organisms that we have altered the DNA of to give them.
We use them for farming.
Yeah, we use them for farming and for industry, for making drugs even. Insulin, for instance, is made in genetically modified bacteria. Did you know that, Brandon? Yeah, insulin, human insulin. We can take the gene for human insulin and we can put it into a bacteria, and that bacteria will make human insulin for us, and we can just harvest it from it. Bacteria are really cheap workers. And so there's all kinds of special organisms. It's not sci-fi anymore. We can make glowing cats. We can make corn that has bacterial pesticides as part of it. And all of this allows for more food production or for more profit, depending on what we're talking about. Okay, the last one we'll mention here is gene therapy. Remember, gene therapy, the idea behind that is if a person has a mutation that's causing them to have a disease, then we should give them, as the solution, we should give them the gene without the mutation. And that's what gene therapy is. So think of it as like the DNA is the drug. The problem is that they have missing information, and so we're giving them the information. And the cell knows what to do with it, and it can, in some cases, fix problems. Okay? Any questions?